are in airway inflammation in the context of chronic airways disease, innate immunity, the role of infection in chronic airways disease, and translational clinical trial design. Here at the Alfred, he's the director of our cystic fibrosis service at a very exciting time to be involved in CF care when there's very large changes taking place. So it's wonderful to have Peter giving leadership in that space. And he also has involvement in the severe asthma clinic and provides support to people with bronchiectasis and other suppurative lung diseases. So Peter today will present on the topic of integrating research into clinical care. Thank you, Peter.
Thank you very much, Anne. I'll see if I can mouse over here. I can at least move it, and then I can keep eye contact with those whose eyes I can see. Well, thank you very much for inviting me to speak today, and particularly to speak at the first of these sessions for the new year. And it perhaps is particularly timely that the topic is very much translational medicine and what it may mean and how it might look. So in many ways, I'm going to start out with what's a very clinically based talk to talk about the values of a more pragmatic approach to trying to introduce and integrate research into clinical medicine. But I think for many of you who are not clinicians, it's really a notification that, look, there's great resources in terms of the clinical workload that happens here and many hospitals around the country, and effectively integrating that into the fantastic cutting-edge science that you're all doing is, I think, an opportunity. And having well-characterised, clinically defined problems and access to patient samples and understanding how that will translate into clinical research or the clinics themselves, I think is really crucial as a way forward. So in essence, that's what I'm going to talk about. Okay, so much of this is really talking about evidence-based medicine and integrating that approach to a more real-world framework. Most of what I'm going to talk about here is being leveraged off some of those pragmatic-based trials. It's important to say that the results from the choose-betweenumab trial that I'll present today was funded by GSK. So I think it's a false dichotomy to assume that research in clinical medicine should be seen in two different facets. And really, for a clinician and for a researcher, it's the same problem with those different aspects that we need to address. Just like Janus was the Roman god of entrances and exits, it's really a two-form process where we're looking at a clinical problem to inform us as to what the priorities should be. And we now have fantastic tools to be able to bring to bear on the clinical problems that are present. And so discovery and clinical care need to come together in order to be able to solve those problems, not surprisingly. So if we think about clinical medicine and clinical research, there was a significant movement that began really in the late 1970s to improve the quality of clinical research and to really apply a scientific rigor to that. Up until that time, it had been largely observational. Clinical trials were poorly defined or had poorly defined protocols. And often the statistics that were starting to develop at this time were not being applied in the correct sort of manner, or the clinical trials themselves, when they were being recruited and designed, had very substantial flaws. And so what was really a revolutionary change in statistics and their application to biological processes that had occurred in the 50s and 60s started to be applied in the clinical context as well. And many of you will probably recognise that's Gordon Guyatt there, who is really regarded as one of the fathers of evidence-based medicine. And it was this concept of introducing that scientific rigour and then applying it to clinical care so that clinicians would use the best available evidence to make those clinical decisions. And they've got this hierarchy of approaches here with evidence-based and highly scientifically rigorous approaches, such as the randomised control trial, being at the top, or rather a whole series of these now undergoing systematic meta-analysis, sitting at the top of the pyramid. Whereas what was regarded as older school observational, opinion-based sort of approaches sit down at the bottom, which is not really the same that none of those things are of value. And from this emerged very important things such as the Cochrane systematic reviews, and then of course now for guidelines, the grade approach to assessing evidence and implementing those into practice. So the prospective randomized controlled trial, you will all know, is the gold standard for clinical research. And it's got enormous strength in terms of its internal validity. So you can establish causation. Randomization minimizes biases, both internal and external, while blinding, of course, also reduces those biases, both from the point of view of the participants, but also from the investigators. And we now have a well-structured approach as to what a randomized controlled trial should be, and even registering these randomized controlled trials before we move into working on them. However, they also have weaknesses. And essentially, the weakness is in this inherently rigorous approach that one takes in order to be able to do a randomized controlled trial. Inevitably, you want to minimize the variability which is inherent in most human populations. And by doing so, you highly select out groups of individuals. You naturally will only select out highly motivated or at least well enough people to take part in these clinical research trials as well. And so how applicable that is to our disease populations can vary enormously. And you can see over on the right-hand side here just a bit of a snapshot from Travers and Beasley from 2007, where they looked at a group of individuals from New Zealand that had been assessed with chronic asthma. And they could find that less than a third would really fit the criteria that were required to allow them to be randomized into an asthma trial. So for a clinician, that means, well, 2/3 of my patients, the evidence that I'm getting, I don't know that I can apply that to the person sitting in front of me. The other important and increasingly important factor is cost. The cost of running these large randomized placebo-controlled trials is significantly increasing, as is the cost of everything. But there's intense logistics, intense time in terms of clinical research offices, and the costs themselves are highly dependent upon the numbers of patients and the number of sites that you need to involve. And an example here is for the last 12 months, the 225 pivotal trials that the US FDA had used to approve a product, around about, over $40,000 per participant had to be spent in order to get those products before the FDA. That is an enormous cost. We have to have results that are externally valid, but we also have to have a way forward where we can have a cost-effective approach to introducing evidence into clinical practice. So there are many barriers, of course, to clinical research, as you know. Funding and infrastructure, I don't need to tell any of you here, is problematic. I worked on that. Workforce. Our workforce is not treated that well. I mean, clinical researchers and all research staff are generally on very short-term contracts. They're not integrated well into health services. And it makes it very, very difficult, therefore, in order to be able to do that research. Participant recruitment has always and always will be a major challenge, but there's costs in both time and money. There's an inadequate knowledge out there amongst consumers as to the importance of that clinical research and the knowledge and understanding of what clinical trials are, and significant fear as well, particularly in regard to placebos and the whole concept of randomization to treatment. There are also significant regulatory constraints that have not gotten easier over the years, but in fact, increasingly represent a very substantial burden in order to be able to execute clinical trials. Now, that's not just ethics, but also now governance, and particularly governance between every institution that is inevitably involved in the process of research is a major barrier to taking things forward in terms of both time as well as costs. So that brings me to this concept of talking about pragmatic trials, and I'll give you some examples as to how this might be used to both able to leverage that research into a clinical setting, but also so that it can be used to support basic science research and researchers into mechanisms of disease as well. So by pragmatic trial, I mean this real world evidence concept, and it's become a bit of a flavour of the month sort of statements to make. Some pragmatic trials are not very pragmatic, but really it's designed to try and broaden this concept of external validity. So a prospective randomized controlled trial can still be performed as a pragmatic trial, but there are some compromises made to make it more generally available. So the main advantage over simply observational data is that you can still use the inherently powerful tool of randomization, but you would apply it in a very light-on approach and, where possible, in a clinical setting, where you minimize the need to reproduce ways of collecting data and access to participants, so that you're really still getting that strong internal validity signal. So an excellent example of this was the recovery trial during the pandemic. It is important to say that last week this was defunded by the NHS, and that says a lot as well, but anyway. So you will know that the recovery trial was an international clinical trial that identified treatments for people hospitalised with COVID-19. Not surprisingly, and particularly for the NHS, this was a crisis of enormous proportions. And the fact that clinical research could be conducted in a system such as the NHS in such an effective way is a real testament as to what can happen when the circumstances are right and the drive to be able to achieve this is also ensured. So they had a trial that was designed to be light on, but for extraordinarily busy clinical staff, they could recruit to the trial and they could record the outcomes. They had a pre-specified protocol that they had ready to go. Fortunately, we had the swine flu pandemic of 2007, which really had that right and ready to apply very, very quickly. They had a streamlined consent process, and they also had web-based interaction to be able to upload people into that and to transfer the outcomes across very quickly. And it worked. So with that right scientific rigor and the correct application to this very important clinical setting, you can see a phenomenal response. And really, this was the source of the major findings for clinical outcomes that could be used to treat COVID-19. And the time frame is just fantastic. So the 10th of March was the first draft protocol was written. The first patient was enrolled 9 days later. By the 4th of May, they had 10,000 patients enrolled. It's helpful if you have a virus that infects a large number of people and perhaps reflects the not so good public health response from the UK. By the 5th of June, they determined that hydroxychloroquine was ineffective, though we still funded 2 or 3 progressive randomised controlled trials looking at that for the next 18 months, but anyway. By the 29th of June, lopinavir, ritonavir, no clinical benefit. But the 16th of June, the first effective treatment for hospitalised patients with COVID was found to be present, and that, of course, was dexamethasone. So within 6 months, we had clinical evidence to back up clinical decision-making. Obviously, an extraordinary period of time, and obviously an extraordinary response as well. But it does demonstrate the strength where this can be applied under the right circumstances. This is also an important area, though, for diseases which are complex and rare, because doing large clinical trials in order to recruit people with very rare disorders is incredibly challenging. And the usual models of prospective randomized controlled trials and being able to have substantial enough people and sample sizes that will yield significant scientific results is obviously often a barrier in order to be able to push treatments forward, or at least get them through the first hurdles of treatment. So there are several approaches to this, and I won't dwell on any of these in greater detail today, and you're probably better off getting some of my oncology colleagues to come and talk about this. But these are examples of applying or reapplying some of the methodology that's present for randomized controlled trials, but in a way which is really designed for rare diseases. And we'll talk about that at the end with an example for cystic fibrosis. The other approach is to use a Bayesian approach for adaptive clinical trials as well, and leveraging the medical statistics and the ability to really play winners in these sorts of outcomes. And again, in rare disease, this can be very helpful. So let me try and indicate where this can be a helpful thing. And we'll start with the clinical example of doing a randomized, pragmatic, real-world trial, and then where research can leverage off the presence of these clinical samples to have some discovery aims, which are always very good along the way, particularly when you have to wait for a large clinical trial to conclude. Your funders like seeing results as they come along. So the Choose Between a Map trial targets people with severe allergic and eosinophilic asthma. For those of you not in the respiratory space, severe asthma has undergone quite phenomenal changes in the last 10 years with the application of monoclonal antibodies or biologic therapies that target very specific components of the type 2 inflammatory cascade. We have agents that target IgE, omalizumab, agents that target IL-5 or the IL-5 receptor, mepolizumab and benralizumab, and also agents that target IL-4 and IL-13 or dupilumab. However, each of these were developed independently. They followed very similar formulas and recruited very similar patient groups. But of course, the goal for pharma was to get their agents out there and available and approved for use, which they have effectively done. But there are no comparative trials. What do you use if your patient can receive one or more agent? And is one agent better than the other, or are they equivalent or at least non-inferior? So this trial set out to do that. We wanted to determine whether patients with both severe allergic and eosinophilic asthma would respond to these two different biologic agents, omalizumab that targets IgE, mepolizumab that targets anti-IL-5. So all of these patients could receive either of these agents under the PBS, but this trial was set up to compare those outcomes. So we set it up as a non-inferiority. It was unblinded, pragmatic, but randomized, so patients were randomly assigned to treatment and then prospectively followed. It was a multi-center trial, and it had really complex inclusion criteria. And you may say, well, doesn't that defeat the purpose of a pragmatic trial? Well, maybe, but these are actually the PBS criteria. And so that fits nicely under these circumstances. And I'm sure many of you can think of examples of this in your own subspecialties where the inclusion criteria can be quite rigid, but as long as you have a mechanism in place. So that gives you a really well-defined clinical cohort, and that has obvious implications if you want to start looking at it from a discovery lens. Our primary outcome was a change in symptom control at 6 months, which was probably the most responsive outcome for this group of individuals, well recognised in the original regulatory randomised control trials, and we had a non-inferiority margin of a change of 0.35. We had a number of secondary outcomes as well, which are important, and I certainly have interim data on that at the moment. And a non-responder was really a non-responder as regarded by the PBS, who wouldn't go on and have treatment. Now, this is really quite a low bar for patients to be able to get across. The changes in symptoms or the reduction in exacerbations were really not too difficult to achieve, if you take my word for it. But we also applied a little bit more of a rigorous criteria. So people can continue on with treatments. However, we wanted to know who was doing really well. And so we've defined this group according to what we call clinical remission. So really good symptom control, a value of 0.8 is very, very good symptom control of their asthma. No use of oral corticosteroids and no exacerbations. So far more defined. You don't have to make that goal in order to get approval of a PBS. So this is an interim analysis. Well, part of it's an interim analysis and part of it's the primary analysis. So forgive me, I've got the primary analysis of the outcome, but not the secondary outcomes. So this is for the interim analysis at the moment, looking at the two groups there. For those of you not familiar with asthma, take my word for it, but It's a pretty typical group of individuals that we prescribe MABs for. They've got a degree of fixed airways disease, they're middle-aged, they have a really high burden of disease with lots of prednisone use and really poor symptom control and frequent exacerbations. So the difference we looked at was after 6 months of treatment, looking at improvement in that symptom score. The ACQ has a minimal clinical difference of 0.5. None of the registration trials actually necessarily show that that's the case, but the real-world data often demonstrates people have an even bigger effect than what was seen in the original registration trials. So head-to-head comparison after 6 months of treatment, there was no difference between the two monoclonal antibodies. People were just as likely to experience an improvement in their asthma symptom control at 6 months if they received treatment with omalizumab or mepolizumab. And this met our criteria for non-inferiority. The secondary outcomes, we're still working through these. This is still on the preliminary data. No reduction, as you could see there, based on an intention to treat process, as well as the protocol, which I just showed you. There was a very substantial reduction in exacerbations, but it was the same in both groups. The failure to respond, probably about the same. More people were likely to switch from omalizumab to another monoclonal antibody, however, which is interesting. And in terms of attaining clinical remission, in fact, the alternative might be the case in that people were more likely to attain clinical remission if they received omalizumab. Adverse events were not really very severe and pretty similar between the two groups. So at this stage, you've got this dichotomy that perhaps more people get a more sustained and reliable effect from mepolizumab, but in fact, omalizumab might have a more dramatic effect, at least for a small number of individuals, though identifying that is really not possible based on the clinical data alone, at least with these inclusion criteria that we've got there. So we now have a direct comparator of these two agents, and we've used a pragmatic clinical trial design in order to be able to say, well, there is actually no difference between the outcomes, at least 6 months, in these two interventions that are there and available on the market, which I guess doesn't help the clinician awfully much, in that we still don't know which agent to give them. So that's where the discovery aspect hopefully comes into things. So the first was we took a subset of individuals and we looked in more detail at some of the immune responses from those individuals. We looked at a type of cell called the innate lymphoid type 2 cell, ILC2 cells, which we know play a very important role in innate immune responses in the context of asthma. ILC2 cells are really potent generators of those type 2 inflammatory cytokines, interleukin-5, interleukin-4, and interleukin-13, but record the inflammatory cascade which is present in asthma. And so what we did was, in a subset of these individuals who entered into the clinical trial and then received treatment for 6 months, we cultured the ILC2 cells from them. This is the group here. We also were able to have some comparative groups so that we could look at that comparison, a group of healthy non-atopic controls, a group of healthy atopic controls without asthma, a group with mild asthma, and then that subgroup of individuals with severe asthma who went on to have treatment. Not surprisingly, the group of severe asthma, the mild asthmatics have really mild asthma, and of course the non-atopics have virtually no inflammation. So you've got really well-defined clinical cohorts that you can look at this at. But now you can also introduce an intervention in your cohort that have got the most severe disease, and that was, of course, the clinical trial. So this is really busy, my apologies for that, but it's looking at some of the markers in the cultured ILC2 cells from the individuals from each of those cohorts. The healthy controls, the non-asthmatic, atopic individuals, the mild asthma group, and the severe asthma group. And when you look at the ILC2 cells from those individuals, the severe asthma group, you can see there in red with the individuals, you've got high expression of markers we know are associated with type 2 immunity, TSLP2. The cells are highly proliferative. In fact, they would culture and grow extraordinarily easily whereas culturing the cells from almost everybody else was quite challenging. And they not only released intensely those type 2 cytokines, IL-5, IL-4, and IL-13, but in fact, that correlated with features of disease control. The IL-5 release of these ILC2 cells correlated very closely with the peripheral blood eosinophil counts of those individuals with severe asthma. It also, well, IL-4 and IL-13 correlated closely with exhaled nitric oxide levels in the exhaled breath of these individuals, a biomarker of IL-13 activity in those individuals. So you can see that taking these individuals and having a well-defined clinical cohort, you can relate these biological responses back to those individuals that are present. We then, of course, went and treated them with monoclonal antibodies. And as you just saw, these monoclonal antibodies work really, really well, although through different mechanisms, in controlling asthma. So we saw that people who received treatment, their clinical disease improved substantially. And the biological effect on their ILC2 cells was also quite profound. Now, all the groups showed an effect, but in fact, in this case, mepolizumab had the greatest effect on suppressing that hyperproliferative response amongst those ILC2 cells, whereas omalizumab had a far more variable response. Now, as you know, omalizumab still worked, but was working in different ways. However, you use the anti-IL-5 monoclonal antibody, and you can see in that biological readout a very well-defined, improved biological response as well. So blood ILC2s from severe and allergic eosinophilic asthma subjects, when stimulated, demonstrated an increased proliferative capacity and an increased expression of TSLP, GATA3 and NFAT2, not NFAT1, not surprisingly, type 2 markers of inflammation. Mepolizumab substantially suppressed that hyperproliferative innate immune response, and you could see that translate to the clinical response in those individuals as well, and the mechanisms by which this was having an effect. So how could this be occurring? Well, we still probably need to dig a bit deeper, and perhaps things such as RNA-seq and single-cell sequencing can inform us. Now, I don't have the single-cell sequencing data yet, but I do have the bulk RNA-seq that I'll touch upon. So, not surprisingly, if you take peripheral blood from that group of individuals with asthma, you see that people with severe asthma have a significantly different inflammatory process compared to the patients with mild asthma and, of course, the healthy controls. And the severe asthma are on the far right-hand side there. Genes are switched on, genes are switched off. There's lots of differences between the groups. No great surprises there. We saw upregulation of lots of inflammatory genes, not terribly surprising either. The ST2 gene is related to an inflammatory pathway we know in asthma. Things such as proliferation of airway smooth muscle and fibroblasts, pretty expected. This was a group with significant type 2 airway inflammation, so click genes are up. SLIPI is an antimicrobial gene and an anti-elastase agent, again indicating the intense inflammatory response there. And also the recruitment of B cells and B cell differentiating factors. The only gene that was down-regulated in the severe asthma group was this gene called EPHA2, which not a lot is known about, but interestingly, it has an effect at antiviral responses and improving the efficiency of the host's antiviral response. And we know that people with severe asthma are more susceptible to viral infections. And when you compare that to the different groups, you see quite significant differences. If you compare the severe asthma group to the mild asthma group, again, you can see a series of genes that might be different between these two groups, although there's not an awful lot of differences once you move away from just the healthy controls. And when you look at the treatment effects of mepolizumab versus omalizumab, as I hinted with the ILC2 data, you actually see quite a different picture in terms of the innate immune response of those cells. So treating severe asthmatics with mepolizumab really switched off those type 2 genes that were present in those individuals. No great surprises there. There was downregulations of lots of these genes with mepolizumab, and that significant suppression of those innate immune responses came through. Now, you'll remember that people did clinically just as well with omalizumab, but it was a different story. So while mepolizumab substantially suppressed the inflammatory genes that were upregulated, omalizumab, to a lesser extent, did the same thing. But in fact, now you start to see genes being upregulated. That wasn't seen with mepolizumab. And the upregulated genes are things to do with immune responses, particularly things to do with immune responses against viral infections. The IFID genes are well known for susceptibility with rhinoviruses and influenza. And this is a somewhat unexpected sort of feature, but something that has previously been described when omalizumab has been given to children with poorly controlled asthma, in whom virus infections are often the most common trigger for acute exacerbations. So you still see a clinical effect, but then when you go and apply a discovery approach, you can see that these agents are working in quite different, if not complementary ways. Of course, you're not allowed to prescribe them together. The final thing I will touch upon quickly is then applying this to a rare disease model. And this is the example of cystic fibrosis. So we know a lot about cystic fibrosis now, and we also have effective interventions for cystic fibrosis. We know that cystic fibrosis is the most common life-limiting mutation in Caucasian populations, and that we know that the abnormal protein, the cystic fibrosis transmembrane regulator, is important in conducting chloride, ions, and hydration of airway mucosal surfaces. And where that is dysfunctional, that leads to organ damages and recurring lower respiratory tract infections. And so it's a nice model to clearly define what's going on. But the genetic abnormalities that cause cystic fibrosis are really quite heterogeneous. And there are over 2,000 genes that can be associated with this. We now have CFTR modulators that directly target the protein. They're small molecule agents that will improve the function of CFTR if there is some protein which is present there, by refolding that protein and improving its function, or improving its function where the protein is expressed on the cell surface membrane but is essentially dysfunctional. It won't help where there is inadequate protein or protein which is seriously flawed and therefore not expressed. And these CFTR modulators are now licensed for use in around about or close to 90% of people with cystic fibrosis. So it's a rare disease, and I'll move on. And really looking at that application in a CF context can be quite helpful. One of the reasons that this may be helpful is that there are a broad number of groups of individuals with CFTR dysfunction, some of whom have been approved for use and some of whom have not been approved for use. And how well this agent works for some individuals may also vary, even within those categories of individuals who have approval. And so for an agent which is very cheap, this might not make a lot of difference. However, this is one of the most expensive medications available on the PBS at the moment. And there are people that have been assigned to treatment in whom efficacy has not been established in clinical trials, and similarly, people in whom treatment is excluded because the genetic mutations have been undefined. And with so many genetic mutations out there, it may not be practical to be able to identify all of them and to know who's responding and who's not responding. And so what we've done is we've taken an organoid-based approach to specifically assess the response to the CFTR protein and to see where that's dysfunctional, now that we can intervene. So if we take an individual who has 2 copies of the delta F508 mutation and significant impairment, we would predict that they've got severe disease. And we know that these CFTR modulators are very effective at improving their outcomes. And so this is one individual here. This is their CFTR responses over on the left-hand side. you can see baseline response is virtually nothing. You treat their organoids with the agent itself, and if they receive Trikafta, which is the CFTR modulator, you see a substantial improvement in their outcomes. If you give this agent to that individual, you can see an improvement in their lung function. If we take another mutation where this is again not so well defined, so delta F508, and a mutation where there is no protein expression, you still see a substantial improvement when you treat these organoids with CFTR modulators. But again, if you look at a broader group of individuals in whom efficacy is not defined, and this group of individuals who have residual CF function, you in fact don't see their organoids responding as well. And yet these two individuals have access to the medication under the PBS and prescribing criteria at the moment. So what we're proposing, and then of course there are the have-nots. So if you look at these individuals, they are currently not able to receive treatment with the CFTR modulators. And yet, when you look at their organoids, they clearly do have improvement in CFTR function. So the ORIGIN trial is an N-on-1 trial where patients' organoids will be tested to see if they respond to the CFTR modulators. They will then move on and receive treatment to determine that that organoid response is going to be sufficient to also see an improvement in their clinical outcomes. And so we've set this up in such a way that the individual gives the specimen, they demonstrate a clinical response or a biological response with their organoids themselves. Those who have a biological response will then enter into the clinical trial arm. They will then be randomized to receive the CFTR modulator in a period of 4 weeks on, period of 4 weeks off, but receiving placebo at the same time. And the assessment of the process will be randomized according to a Bayesian framework. So that individuals still get randomized, they still receive placebo, and we have outcomes that will be clinically meaningful and we hope can link to their organoid responses. So they're just some examples of how we might be able to better utilize clinical patient populations, well-characterized individuals, be that in a large clinical cohort or a small, well-defined rare disease clinical cohort, and really involve discovery research in that process to better answer the questions as to what the problems are or what the biological basis for those problems are, but also how our treatments are working and why they are effective and in whom that they may work best. So I'll end it there. Thank you.
Thank you very much, Peter, for that great talk, covering a lot of ground there. We're now open for questions. Going first to the room. I'm curious, as a researcher, obviously, the ILC2 work in your Choose-a-Mab trial was very fascinating to me. But I was wondering about the clinical or the clinician perspective. The omalizumab and the mepolizumab have very similar clinical outcomes. But do clinicians, how interested are they in the sort of fundamental mechanisms of those responses?
Well, I mean, I think it's important to understand the fundamental mechanisms because that will eventually lead us back to deciding how more effectively to use these agents. So I think there's messages in that that we haven't gotten to quite yet. Most of the effects for omalizumab were in fact probably on improving antiviral responses or the efficiency of antiviral responses, which is probably why it does reduce exacerbations. And yet it still does have a high-end effect on adaptive immune responses and IgE responses. And in different sort of settings, such as urticaria and even allergic rhinitis, it's having an effect, which has got nothing to do with antiviral responses. So targeting people in a better and more informed way, I think is fairly effective. I mean, most people will respond to these agents, but the question is, have you given them the best agent and is it the most effective response? And really, we're not answering that scientifically. We just do clinical trials within the broader sense of the term, and the outcomes that we're assessing are very, very blunt tools. And you'll see a lot of people swapped, and there are other drivers of that, such as the complexity of the dosing, the frequency of the agent, the fact that one agent has a nice convenient device where you can self-administer it and the other doesn't. And that's probably not right. We want it better informed. So I think it's looping back to better define who are going to be respondents and who are not respondents is really what I see.
Thank you, Peter. Perhaps I'd start by saying that the way we frame the question actually sets up a lot of our thinking. So perhaps if we also think about how we integrate clinical care into research, then the question leads us to a different pathway. And for the question that just happened, one of the things that fascinate clinicians is the paradoxical responder, the outlier, what's underlying that. And that can often be the tip of the iceberg of a whole different way of thinking. Because a lot of these studies are set up with a particular model in mind. And for instance, the omalizumab MIPO assessment, if we had thought of, could there be an outcome in terms of how frequent or how severe viral infections were, and the length of follow-up was significant, that could have led to a different type of assessment. So I think integrating clinical care into research is also another way to think about it, and they're not contradictory, they're not mutually exclusive. They're complementary. I'd be interested in your thoughts.
And look, the whole process of clinical trials is designed to make sure that the outliers get into the clinical trial in the first place.
That's why it has to be complementary.
And yeah, and I think that's important because there is often significant information found in the outlier, be that either a super responder or a non-responder. Because when it gets applied in a clinical setting, those people will receive that medication or that intervention, whatever that may be. And understanding why it's succeeding or why it's failing is equally important.
Taking the mic back. Thank you for some interesting stuff. So my question is a little bit conceptual. You've compared omalizumab and mepolizumab, but in this space, there's other drugs, monoclonals, coming into the space. So I don't think it's licensed yet, but dupilumab is
It is licensed.
It's licensed now? Okay. So how do you take the outcomes of these kind of head-to-head trials and then integrate them with the literature of other drugs that operate by different pathways and perhaps, I guess I would argue, potentially more likely to have a positive outcome for things like dupilumab? How do you compare them with that? Do you have to set up another trial?
Well, I guess the short answer is probably yes. I mean, it's interesting, and there's bias out there as to how these agents work, and that one of these agents will work better. I mean, the most difficult thing about that clinical trial was that people didn't want to put anybody in the trial because the perception of clinicians was that mepolizumab worked much better. And maybe it does, but not according to the outcomes by which we're measuring it. And so, I mean, that in itself is a message. And similarly, dupilumab, without a head-to-head comparison, we don't actually know whether it works better or not. And it may, but you don't have that. It is possible that you could run a prospective randomized controlled trial with all 4 of the agents that are available there now. That is feasibly possible, but you have to collect the data and then analyze it. Is it worthwhile? Well, it's a question to ask. Could you identify who's going to respond to one rather than the other, and therefore save time and effort of going through a fruitless 6 months? That would be desirable as well. Lots of factors come to play there. It's feasible to do, but there has to be the will to do it. And that will probably isn't going to be driven by pharma.
Can I ask, was there any preclinical evidence to suggest it was going to be better? Or was it just, it's not going to kill people, therefore we'll try it? Like, what's the...
Well, we know individually, well, I mean, we knew individually that they would work because they had both been licensed against placebo. in order to be able to succeed. But is one better than the other was really, is one better than the other? It wasn't a superiority design, it was a non-inferiority design.
Yeah, maybe following on from that. So has that study changed behaviour amongst clinicians, and how does one go about propagating the information you generate in your study to become widespread practice?
Well, we'll present it at the ERS, which I haven't done yet. Will it change practice? I mean, probably not. I mean, people who had omalizumab was the first of these agents to become available, and then the others have come in consequent to that. And there's, the shiny new toys get a lot of interest, unfortunately, which is a little bit sad in many ways. I mean, other messages to come out of that is that there are probably complementary mechanisms going on here, and perhaps we need to think about our approach and who we're targeting more than anything else. The antiviral stuff, can we better characterize individuals going into it, or should we use a combination approach with these agents? Would that be more efficacious? I think they're the types of questions that it probably generates. But in terms of that head-to-head comparison, I fear it's probably not going to change things dramatically.
So having said that, so as a clinician, do you think you can actually control big pharma? Like, do we need 25 monoclonals for TNF, for example? And if you refuse to use it, if it's not suitable, then maybe they'll actually put their efforts into other diseases.
So just for people online, because you don't microphone, you agree with the question.
Oh, do you think that we can change big pharma? Well, by highlighting this issue. I mean, I think the essence of the question is when you start to demonstrate the information and the importance, and I think we have to then drive the agenda. There's a very powerful agenda for superiority trials to get single agents up. And look, not wanting to, I think it's probably wrong. It's very expensive to get these agents up and to get them across into clinical trials. It would be incredibly hard to do comparative trials, and there's no financial incentive to do that unless you think your agent is superior, essentially. Or you just want to get something registered in which you're just saying it's non-inferior. But that gets driven also by regulatory authorities. However, completing that loop and finding different mechanisms and better identifying people, then that gives us the option to go back. So hopefully it then does start to determine people's behaviour. But also, we now have agents and interventions that are highly selective. And in the past, we would just give people prednisone, and it was a very blunt tool that did many, many things. These agents are really highly targeting these different inflammatory pathways. And we can start to tease apart what happens and what's redundant and the mechanisms by which they're working in improving the clinical outcomes. And that itself will hopefully inform us as to new treatment approaches to this. I mean, those down-regulated and up-regulated pathways are all worth exploring further.
Microphones, we're going to Margaret first.
Robin and I have been having a discussion as this discussion's been going on, but we were both wondering why on earth can't you combine these two antibodies, given they are targeting different pathways? And is it purely that they're owned by companies, or why can't you do that?
So you can't because you're not permitted to under the PBS. The government won't pay for it. And no, nobody has taken that and moved forward with it. You could, but again, you've got to answer the question, well, would that actually be meaningful? And it's probably not necessary in the bulk of people with severe asthma. However, there might be subgroups. So I think more work would need to be done to define that. But look, the mechanisms by which they are working, not surprisingly, are complementary. And that in itself is probably informative. It might allow you to better target individuals, but yeah, it is theoretically possible. You could combine these agents. Why not?
Thanks, Peter. That was great. Two questions. One is, I really like the the title of your trial, Choose Between a Map. Fantastic. I remember that. But at the end, did it really provide data to help the clinician choose between these two antibodies or not? I mean, there's lots of really interesting mechanistic data to discuss, but as a clinician, is it helping me choose between a map?
Well, at the moment, I mean, I think it demonstrates non-inferiority, and that's probably in its own right. So that few people are prescribing omalizumab now. Will it change clinician behaviour? Well, that's a harder thing to do.
But does it actually give you information to inform changing clinician behaviour? Does it help me decide how to choose between these two drugs?
Not yet. I think that's the step in the right direction. And as you probably appreciate, most science is incremental as opposed to fundamentally changing. No, it hasn't fundamentally changed things. But having a good acronym is very important.
Very good one. I love it.
High impact journal, yeah.
So my second question was, I absolutely agree with you in terms of the advantage of pragmatic randomised trials being more real world applicable and achievable, and and comparing between drugs that are licensed. Another approach that Helmut Scruben, I don't think he might be online, has led within the neuroscience field is doing registry-based trials, where he set up this very large international MS base, which now has enrolled prospectively over 100,000 patients across 40 countries. And they've used that to compare various licensed treatments in a sort of pseudo-randomised controlled trial type design, confounding. And it's provided really informative, high-quality data of drug X versus drug Y for multiple sclerosis. Did you want to comment on that approach and the strengths and weaknesses? And is that another way we can move the science forward quicker?
Yeah, I didn't talk about registry-based trials, but look, yes, that is a really untapped resource. So in terms of cystic fibrosis that we mentioned, we actually have a fantastic, well-resourced, comprehensive registry of patient information with really critical patient outcomes that are recorded in real time. And the integrity of that data is very, very good. It's the perfect example of where you could apply a registry-based clinical trial, particularly now where the game has changed in CF. So short-term effects such as stopping exacerbations or preventing declines in lung function, you're not going to see that in any intervention that occurs over a 6-month period any longer, unless people are not able to access modulators. If you're wanting to look at the effect of withdrawing a treatment or the impact on pregnancy and safety, then the registry has the ability to be able to do that. At the moment, the Australian CF registry hasn't run a registry-based trial, but we're in the process of looking to be able to do that, and we think that that's possible. An example that we think we're going to lead with is actually taking treatments away component of things and doesn't matter to their outcomes. So frequency of appointments and things like that. So registry-based trials, particularly for severe diseases or where outcomes need to be followed for a very long period of time, ticks all those boxes, particularly of integrating real-world data, but collecting that data in a way that doesn't require an onerous or expensive way of collecting outcomes, etc.
Time frames to answer questions are so much shorter, aren't they?
I'm going to take the liberty of the last question as the chair. And I was struck by the difference between the data you presented in asthma and the data you presented in CF, because in CF that really is an example of personalised medicine, isn't it? That's for the individual. And in asthma, we could see that there was some variation in responses, particularly with omalizumab. So how close do you think we are to getting to personalised medicine in severe asthma, and what would it take to get there?
So that's where I think you need this data about understanding that all severe asthma is not the same. And in fact, this is a cohort of what we would regard as a highly defined group of individuals with severe asthma. I mean, they're really super selected. Now, they're a fairly large group of severe asthma, probably about 80%. That means 20% are not going to fit this bill. But even there, you can see the heterogeneity that's present. So I think you've got to understand the process better. For some of those individuals, there were different drivers of their disease process going on, and that's where the discovery aspect comes into it. So we've taken what we think is a highly defined clinical cohort, and it is, but we've gone back and applied that discovery lens to it and found that there's more to it than just that. And that's the sort of thing that I think needs to then be followed up by further work.
Thanks, Peter. Please join me in thanking Peter for a fascinating.
Watch outside.